Hi, I'm Daniel Bly, a PhD student in the Foa and Gasparini lab at the University of Tasmania. In this presentation, I'll be presenting a brief summary of the findings from my recent PhD thesis, where I investigated a novel human mutation in the protein HOMA1 and studied how this mutation may influence HOMA1 function in neurons or nerve cells. HOMA1 is what we call a scaffolding protein, meaning that it is responsible for binding and positioning other specific proteins and facilitating signaling pathways which involve them, such as synaptic communication, which occurs between a pre- and a postsynaptic compartment. HOMA1 is predominantly located at the synapse, and it is known to be required for a variety of synaptic functions, including the formation of synapses, otherwise known as synaptogenesis. My thesis focused upon a specific variant of HOMA1 called HOMA1BC. An important feature of HOMA1BC is that it forms tetrameric structures composed of 4 aligned HOMA1BC proteins. This is achieved through interactions between the 4 proteins at a region of the protein called the core domain, which you can see on the right. These structures are known to be important in order for HOMA1BC to properly scaffold other proteins. I hypothesized that a recently described human HOMA1BC mutation, R297W, which is located within the core domain, would cause the mutated form of the protein to improperly form tetrameric HOMA1BC. I further hypothesized that this impairment would disrupt the scaffolding functions of HOMA1BC and thereby impair its functions involved in the formation of synapses. During the development of the nervous system, presynaptic connections are formed through contacts between different neurons at structures called axons and dendrites. Axons extend and can travel great distances to reach specific targets and form connections with other cells. Extending axons are tipped by a structure called the growth cone, which senses guidance cues in the environment and uses these to steer it towards its synaptic partner. I sought to test how the mutant HOMA1BC variant affected the responses of growth cones to these cues, in order to understand whether it could impair the connections formed between neurons. I subjected neuron growth cones to a motility assay, which involves the creation of specific gradients of a guidance cue on one side of the growth cone and measures its response to it. Within this assay, the cue that I used induces growth cones to turn approximately 10 degrees towards the source of the cue. I found that neurons which contained R297W, but not the wild type or normal HOMA1BC, displayed aberrant turning responses. Where growth cones should normally turn towards this cue, neurons containing the mutation turned away from the cue, suggesting that it disrupts the guidance of axons and could therefore impair the formation of synapses. The synaptic compartments which form connections with axons are called postsynapses, located on the dendrites. The dendrites of neurons are studded with structures called spines, and it is here that the synapses are located. As spines form stable synaptic connections, they mature, and their morphology changes. Spines which have wide heads and thin necks, for example, generally form more stable and stronger connections than those spines which are long and thin. Thin and long spines with weaker connections frequently degenerate and reform, altering the overall density of spines on a dendrite. I characterized the morphology and density of spines in neurons containing either normal HOMA1BC or the HOMA1BC mutant R297W. I found that neurons which contained the mutation exhibited more immature spine morphologies with longer but thinner spines, but an overall reduced number of spines as compared to normal. I extended this work by tracking the development of spines in live neurons over time, taking measurements of their dimensions and density when they were 4 and 8 days old. Here again, the density of spines in mutant neurons was consistently decreased as compared to controls. I found that the spines of neurons containing normal HOMA1BC shortened in length and constricted around the neck as they aged, while no change in dimensions was observed in mutant neurons over the same period. This indicated that spines within neurons expressing the mutation were slower to mature than normal. The regulation of calcium signaling, involving the transport of calcium ions to different areas in and out of the cell, plays an important role in regulating both axon guidance and the formation of spines. I hypothesized that the effects of the mutation described thus far could stem from impaired calcium signaling. I was particularly interested in a process called store-operated calciumentry, or SOCE. This process involves the influx of calcium ions into a cell following the depletion of calcium levels within internal stores in that cell. I measured SOCE in both growth cones and neuron cell bodies with live cell calcium imaging. I found that the amount of calcium flowing into neurons during SOCE was significantly blunted in mutant neurons as compared to their controls. This was true both within the growth cones with which I conducted the axon guidance assay and in the neurons where I examined spines. Next, I was curious to see whether the mutation may disrupt other forms of important calcium signaling processes. I elected to measure the calcium release from internal cellular stores called the endoplasmic reticulum, or ER. We know that HOMA1-BC is important for scaffolding calcium channels on these stores and linking them to a receptor called metabotropic glutamate receptor 5, or mGluR5. HOMA1-BC tetramers in particular are necessary to link the activation of these receptors to the release of calcium from the ER. I used a pharmacological activator of mGluR5 to induce calcium rises in neurons and recorded the resulting signals. I found that neurons containing the mutation release significantly less calcium compared to neurons containing normal HOMA1-BC. This suggests that the mutation uncouples a link between internal calcium stores and mGluR5 receptors. This particular form of calcium signaling is known to be important in regulating how neurons communicate with one another at the synapse. I hypothesized that the impaired scaffolding function of the mutant HOMA1-BC was causing both the ER and mGluR5 to be improperly recruited to spines where the postsynapse is located. I used specific markers of the ER and mGluR5 to visualize them within neurons and compared their presence or absence within spines. I found that the mutant neurons contain significantly less ER and mGluR5 within their spines, confirming that the mutation reduces the recruitment of these two key components to the synapse. As a scaffolding protein, the functions that HOMA performs are achieved through its relationships with other proteins and how it binds and positions them. I next sought to investigate how the clustering and scaffolding of important binding partners of HOMA1-BC might be affected by the R297W mutation. To achieve this, I had to use a particular form of microscopy called super resolution microscopy, which allowed me to visualize different proteins separated by about 10 nanometers in distance, or 1/1000th of a meter. With this technique, I characterized within spines of neurons the density of clusters of several proteins involved in regulating the calcium signaling processes which I had earlier investigated. In each case, the density of the clusters within the spine heads of mutant neurons was significantly decreased, suggesting that the mutant HOMA1-BC is unable to scaffold and sequester these important proteins properly. Super resolution microscopy gives us such a level of detail that we can effectively characterize the relationship between all of the HOMA1-BC in a particular region of a neuron and the proteins which it scaffolds. In growth cones, I characterized how closely coupled in space normal HOMA1-BC and mutant HOMA1-BC were with mGluR5 and another important protein in store-operated calcium entry called STIM1. Mutant HOMA1-BC was significantly decoupled from both proteins as compared to normal HOMA1-BC, which suggested that its interactions with and binding to both proteins was impaired by the mutation within its core domain. In summary, this project has revealed that R297W, a human de novo HOMA1-BC variant, disrupts a variety of important HOMA1-BC functions in the formation of synapses by neuronal growth cones and dendritic spines. Because the mutation differs from normal HOMA1-BC by only one substitution mutation within the core domain, these findings suggest that this region is necessary for a wide variety of HOMA1-BC functions. I hypothesized that the mutation impairs the capacity of the mutant protein to form tetramers, and that the mutant protein can also disrupt the formation of tetras by normal or wild-type HOMA1-BC. This change would be expected to substantially disrupt the ability of HOMA1-BC to perform the scaffolding functions at the synapse which it's responsible for, and plausibly explains many of the impaired functions brought about by the mutation which I have shown to you today. I'd like to thank the NHMRC and the HOMA-HAC Foundation for their generous funding of this work, and my group members and supervisors Lisa and Rob for their contributions. I'd also like to thank you for watching.